Thank you, everyone. So at first, I would like to thank CCC Conference for giving me this opportunity to present here. And the topic today is clinical utility of integrated genomic profiling in pediatric brain tumor. So why we cared about pediatric brain tumor so much? As we know, pediatric brain tumors are the most common solid tumor among the children. They actually represent 20% of childhood cancer. And the prognosis and the therapeutic options varies dramatically depending on the tumor types. So of course, there are biologically diverse diseases containing various kinds of genomic alterations, such as SNVs, CNVs, indels, and gene fusions. So at CHOP, we developed comprehensive solid tumor panels to interrogate those tumor samples. So this panel actually contains two subpanels. The first part will be the solid panel. It covers 238 tumor-related genes, and it investigates the DNA level to identify the SNV, indels, and CNV in one single NGS sequencing reaction. On the other hand, the fusion panel, it covers 110 major fusion partner genes, and it can detect up to 600 already known fusion, and also other novel fusions involving these partner genes. So the combination results from these two subpanels will be set out by our directors. Since we launched our test in January 2016, we have investigated 222 brain tumor cases in a period of 2 years, roughly 2 years. So most of them are ordered with comprehensive tests, and most of them are primary tumors. The mean age of our patient is about 12 years old, and about 90% of patients are actually younger than 20 years old. And if you see for this graph on the right-hand side, we distribute our patients based on their ages and genders. So we kind of see there is a slight increase in the number of male patients, and also the diagnosed age in the male patient is slightly increased too. So we have identified more than 26 different subtypes of brain tumors in our patient cohorts. This pie chart just shows you the 4 major categories we identified. They are astrocytic and oligodendral glial tumors, followed by embryonal tumors, and the mixed neuronal glial tumors, and eventually the ependymoma tumors. And these 4 categories can be further divided into different subtypes, and I'm not going through every one of them here. And we also employ the somatic variant interpretation system proposed by Dr. Marilyn Lee and her colleagues for our variant classification. And I think everyone here should be very, very familiar with this guideline, so I'm not going into the detail. For that being said, we can identify the clinically significant variants in 93% of our cases. That includes 135 cases positive with tier 1 or tier 2 SNV or indel, 136 cases positive for CNV variants, and also 64 cases positive for fusion. And as you can see here, many cases are positive for more than one kind of variants, and especially there are 9 cases actually are positive for SNV, CNV, and gene fusion. So if we look closer to these 3 different groups of variants, we will find that BRAF, TP53, and F1, and H3F3A genes are 4 most commonly mutated genes for SNV and indel. And the most commonly seen CNV in our patient cohort actually is a gain in chromosome 7q, gain in chromosome 1q, and a gain in chromosome 7p. And again, this highlights BRAF here. In this table, I just displayed all 19 fusions identified in our brain tumor patients, including those 12 novel fusions, which have no or very little literature report at the time of diagnosis. And again, I want to emphasize the abundance of KIA1549 BRAF fusion here. So if we just take this for granted, we would think, okay, BRAF mutation is very common in the pediatric brain tumor. But we all know brain tumor is a highly heterogeneous disease. So we think it's better to dissect the molecular characteristics according to different subtypes. So for pilocytic astrocytoma, which is the most commonly seen pediatric brain tumor in our test, we found that 15% of patients are positive for BRAF, SNV, or Indel, especially for this famous V600E mutation. And also 20% of patients actually are positive for chromosome 7q again. Again, BRAF appears here. And also over 50% of patients are positive for KIA1549 BRAF fusion. On top of that, actually additional 6% of patients harbors novel BRAF fusion. So in overall, pediatric, like parasitic astrocytoma patients are kind of enriched with BRAF mutation. And diffuse midline glioma is a new entity in the WHO classification 2016. The patients falling into this group actually are 100% positive for H3K27M mutation. And on top of that, over 50% of patients have additional chromosome 1q gain or amplification. So there is an amplification for these H3 genes here. And for the atypical teratoid rhabdoid tumor, ATRT, this subtype of tumor is featured with chromosome 22q loss or copy-neutral LOH. And one very important tumor suppressor gene, SMARCB1, actually located in this region. And 50% of patients actually have additional SMARCB1 mutation. And as you can see here, these mutations are not necessarily recurrent. And here, this is just an example for the ATRT patient. We can see the CNV is respectively clean, but we can clearly see there is a heterozygous deletion for chromosome 22. But if we look closer, we will find there is a SMARCB1 exon 1 homozygous deletion in the tumor sample. And we also perform the MLPA in a blood sample. And this time we only see a SMARCB1 exon 1 heterozygous deletion in the germline. So we figure the patient lost the normal copy of chromosome 22 during the process of oncogenesis and eventually end up with a biallelic loss function of SMARCB1. Eventually, we come to medulloblastoma, and we already know medulloblastoma can be divided into 4 subgroups according to the genomic features. So the sonic hedgehog activated medulloblastoma patients are actually pretty rare in our patient cohort, but we do have 3 of them. And all of them are positive for PTCH1 loss function mutation, and the PTCH1 is a known critical repressor for sonic hedgehog pathway. And also, they are all positive for loss or copy-neutral LOH of chromosome 9q. For the Wnt-activated medulloblastoma, a lot of patients has a mutation in CTNNB1, which encodes beta-catenin protein. And also, loss of chromosome 6 is also very common in this subgroup of medulloblastoma. And non-SH non-Wnt medulloblastoma, also known as a group 3 or group 4 medulloblastoma, they have a pretty diverse SNV genomic profiling, but they do share some CNV features, which is isodicentric 17q. And we also found that in the group 3 and group 4 medulloblastoma, we have like almost 2-fold as many male patients compared to the female patients. So overall, like what does our results affect patient care? So we can say our result changes or specify the diagnosis. For example, if we identify this famous K27M mutation, we will suggest, okay, this patient may have diffuse midline glioma. And then if we found this C11 or IF95 RARA fusion, this patient may have a RARA fusion positive ependymoma. And it can also help with prognosis prediction. For example, TP53 mutant MECAN amplified medulloblastoma usually has a poor prognosis. And IDH mutant anaplastic astrocytoma usually has a better prognosis compared to the IDH wild type ones. And sometimes the treatment suggestion could also be made according to our findings. For example, the patients with a TERT promoter mutation, this clinical trial for IDH wild type TERT promoter mutation, grade 2 or 3 gliomas might be available for them. And for patients with high grades or relapsed tumors positive with BRAF V600E mutation, also there are phase 1 or phase 2 clinical trial might be available. And we also can make suggestions for further testing, for example, for those germline testings. So the table here just summarizes the clinical impacts of our molecular finding in our patients. And I just want to highlight here, there are 6 cases with their diagnosis completely altered by our molecular findings. So in these sections, I would like to share with you 2 case examples here. The patient 1 is a 12-year-old male. The clinical indication is malignant glioma, and a suspect for the diffuse midline glioma. And the comprehensive solid tumor panel was ordered. So at the first glance of this tier 1, tier 2 SNV we identified here, we didn't see H3K27M mutation, so we kind of rule out the possibility of diffuse midline glioma here. And on the second look, we actually found there is IDH1 mutation and also ATRX nonsense mutation as viewed in the IGV here. So many studies have demonstrated that both high-grade oligodendroglioma or astrocytoma will be negative for H3F3A mutation, but positive for IDH1, IDH2 mutation here. And furthermore, the ATRX loss mutation is enriched in astrocytoma, but not in the oligodendroglioma. So this patient might have diffused or anaplastic astrocytoma here. And then we also noticed that there is an actual high amount of tier 1, tier 2 mutation in this patient. Also, he also has like 26 additional VOUS SNV, which I didn't list here. So with this hypermutated tumor burden, we are totally not surprised to see the mutation in TP53 and the MSH6, which are both critical components for DNA repair machinery. We even go investigate these mutations in the blood sample to confirm that only MSH6 mutation is germline. And the classification actually is changed to reflect this feature. Our CMV profiling also provides a series of CMV variants identified here. I just want to highlight this subtle CMV in chromosome 2p here. This is a copy-neutral LH for this region, and this region actually covers MSH6 gene. We also identified the copy-neutral LH on 17p, where TP53 locates. Most importantly, we found the homozygous deletion of CDKN2A, 2B on chromosome 9p. Why it's important? Because we know when the diffuse astrocytoma advances to higher grade anaplastic astrocytoma, it's usually coupled with CDKN2A homozygous deletion or hypermethylation. So we think this patient has an anaplastic astrocytoma. So we also identified a novel fusion, which is KMT2E MATs in this patient, and we single confirmed this fusion too. This is a novel fusion, and is consistent with chromosome 7 deletion observed in our CMV profiling. The amplification or overexpression of MATs oncogene actually has been shown to associate with multiple human cancers. And this PRZ1 MAT fusion, apparently this one is very popular because it has appeared in multiple talks in this meeting. So this fusion has been reported in 1 anaplastic astrocytoma patients and also in the secondary glioblastomas. So in general saying, all pieces of evidence are in line with our conclusion. So in summary, this patient has anaplastic astrocytoma, IDH mutant, and he also has a germline homozygous MSH6 mutant due to the absence of heterozygosity. So in other words, he has a constitutional mismatch repair deficiency syndrome. And based on this hypermutated tumor features, we will say the immunotherapy might be applicable if this patient does not respond to current therapy or the disease relapses. And for the case 2, actually I borrowed some slides from our pathology, Dr. Santi here, and I just want to recapitulate this case from the angle of a pathologist. So as we can see here, this is a primary diagnosis according to the histology. Of course, the integrated diagnosis is pending because the molecular information is pending. And based on the histology, the classification is concluded as an anaplastic astrocytoma, which is WHO grade 3. And then we performed comprehensive solid tumor panels, and we found 5 different tier 1 or tier 2 SNVs here, and including this symbolic H3K27M mutation, and also activation mutation in PI3KCA, and 2 mutation in P10. We also get a pretty clear CNV profiling here, highlight with amplification of chromosome 1q, which contains H3F3A gene here, and also have amplification for whole chromosome of chromosome 2, where MYC-N is located. And also we found there is a loss of 10q here, where tumor suppressor P10 locates. So here comes the final diagnosis. With all the molecular information present here, the integrated diagnosis becomes the diffuse midline glioma, H3K27 mutant, and which is a WHO grade 4 tumor. So I like the comment Dr. Sante put here. WHO classification of CNS tumors 2016 updates breaks with the century-old traditions and incorporates well-established molecular parameters into the pathology classification. So I think this is a pretty good summary of our job. So in summary, I would say we studied 222 brain tumor cases. Most of them are pediatric brain tumors using CHOP comprehensive solid tumor NGS panel. And we can identify clinical significant variants in 93% of our cases, and our results provide genomic evidence for tumor diagnosis or even change the original diagnosis. The potential changes to the treatment strategy based on the risk assessment or target therapies happens in 28% of our patients. And eventually, genomic profiling plays more and more important role in pediatric brain tumor clinical management, and also in understanding the molecular characteristics of different subtypes. And the knowledge we gained here will in turn helps us to develop the new drugs and eventually finding the cure for pediatric brain tumor. So at last but not the least, I would like to thank everyone in the DGD Cancer Genomic Diagnosis team for their contribution to this project. I also would like to thank Dr. Marilyn Lee for giving me the chance working on this exciting project. I also want to thank everyone in the bioinformatics team, GC Core, and other DGD members for their help. And eventually, I want to thank physicians and the staff in the CHOP Cancer Center and the pathology department for their support. With that, I'm ready for the questions. Thank you.
Okay, so we have time for 1 or 2 questions.
Very nice. Can you tell us, for the novel BRAF fusions, how often did you see a copy number alteration on the chromosome that had the partner gene involved?
Well, to be honest with you, we're trying to couple the CNV analysis with the BRAF normal fusion, but it's not very common we can see that. Because we do our CNV based on our NGS data, and this is not a whole genome. So our probe density can vary depends on the region. Even sometimes we identify those common KIA1549 BRAF fusion, but we cannot see it for sure in the CNV analysis.